topic over here is the value of the genetically modified drugs for most parents. The birth of a baby is followed by a year of play, amazement at the way their children grow. Obviously, when a new individual is growing, they will feel happy to notice naughty things. But some children grow slowly. They lack GH, which is the growth hormone, or somatotrophin hormone. The hormone is responsible that is secreted by the pituitary glands, and it would be stimulating growth. Now, for example, doctors overcome the problem by giving children regular injections of pituitary extract made from the pituitary gland, taken from the human dead bodies. Okay, those individuals who have died, they are going to extract the GH from those individuals, and then those hormones will be inserted into the newborn, basically. Although that is expensive because it need lots of money and technology to extract the material from the body of those dead organism, and then to insert into the new one. That is a really expensive technology. So genetic engineering has solved the problem of supply. आप simply individual के DNA को replace कर दें, उसमें changings induce कर दें, तो ये हो सकता है. However, solution came too late for some unfortunate brain tissue from individual who had died as a result of a very rare Creutzfeldt-Jakob disease, CJ. Okay, it's a fatal disease of brain when the person, the disease was linked to the bovine spongiform encephalopathy, BSC, or mad cow disease in cattle. Sadly, some people were treated, some people died because obviously their bodies lacked the growth hormone, and they died. They were dying of it. Okay, next thing is okay, so people sometimes express concern about the risk of using genetically modified organisms in or medicines or food. It is what can come from wide variety of the resources. However, some can reduce. It is quite obvious where some individuals are reducing risk of being jam damage, risk of being having threats of having new DNA or extracted DNA until unless that is extracted in a pure form. So that is fine, but in other case they are going to make the DNA polluted. That polluted or contaminated DNA is not affordable, not acceptable by the body, and that is going to make negative changes, and those people have to risk their life. Next point is banana vaccine. Scientists have successfully used genetically modified organisms to make a certain protein such as insulin, somatotrophin. However, prokaryotes do not possess biochemistry to make some of the more complex human plants proteins. Basically, for this purpose, scientists are working to induce a useful human gene into the eukaryotic cells, such as yeast, plants, and other humans. So now they are modifying the genetic makeup, like whether any gene could be inserted into the transgenic plants or not. Transgenic plants are those plants who are able to carry the DNA, who are able to carry some makeups onto it, and then they are transmitting it. Okay, certain vaccines in a very effective way of eliminating serious diseases. They are less economical. Obviously, if you insert the DNA into a plant cell, there is no harm of it. You can simply insert the DNA into the body of plant, and plant will automatically. There are two results of it: either plants will grow, or either those plants will die. These are the main hardly effect. But instead of risking the life of a living organism who is living organism, eukaryote, prokaryote, is not good. So let's sacrifice plants or fruits even. Okay, next thing would be there can be cultural or practical difficulties with seeing the healthcare workers and allowing them to vaccinate children. Okay, there is another trouble when people used to, even in underdeveloped countries, when people give their babies to general or least trained people to inject themselves with the vaccine. That would lead to cause issues. Obviously, sometimes the injections are not hygienic, that led to cause diarrhea, hepatitis. or any other infections. Sometimes, if they insert that injection in a wrong place, probably on the hip muscle, there is a vein, there is a vessel called as the you call as I think sciatica. So that might lead to the sciatic pain. Even there is a vein, if injection got into that particular blood vessel, that is going to make that children paralyzed throughout the life cycle. So it's not fine to give your baby to anybody to insert the injection into him. Now, local communities could grow relevant plant. Okay, back to the topic. So they grow a relevant plant cheaply, and they no need for extra storage. So those areas where banana trees are growing on a very rapid rate, we can use those area for the plant growth. The children can be protected from the deadly diseases with no need of trained health workers. I mean, like if somehow some genes are needed, we can insert those genes. We can insert nice gene into those fruits, and when people are going to eat it, they can have it. Then, okay. Transgenic genes, making of or formation of the transgenic gene is what the bacterium, agrobacterium, timifications, is usually used to introduce gene from one type of plant to another, or even from one animal to the plant. So when you insert gene from animal to the plant or plant to plant, this is happening in the transgenic plants. So this certain individual causes tumor in plants, which are known as the crown gales or crown galls. It also contains plasmid, just like we have studied in bacteria, that bacteria contains plasmid into it, and if we modify the makeup of bacteria, we can reset that plasmid, we can insert the new DNA. Yes, exactly. Now simply, it contains a plasmid called the Ti plasmid, which transfers bacterial genetic information directly into the plant. There is a tumor present in a plant that causes plant cells, but modified plasmids can be used to carry out the beneficial genes into the plant genome. Obviously, it is going to carry certain gene, and then they are transmitted or inserted into the new plants. But so the grown species, the grown individual, or even that same plant with the modified makeup will carry extra or modified or developed characteristics into him. Then, by the process of plant cloning, we can use the modified transgenic cells to produce a new transgenic plant. So every part of that cell, every cell of that plant will accept the new DNA. And now they will be modifying their genetic makeup. One in the following fig, there is a plant, and in the plant there is a Ti plasmid into it, which was inserted. Okay, or even let's say we have extracted it. So the Ti plasmid is extracted from a certain plant species, and now once it is extracted out of this plant species, the gene to be carried into another plant will be inserted, and then returned to the bacteria. Obviously, there is a new gene that we have just extracted from this plant. Now it is ready to be inserted into the bacteria. Okay, in the stage number 3, the plant is infected with the modified bacterium, and part of Ti plasmid with the genetic engineering genes became part of the plant chromosome. So gradually, the DNA of the new plant will accept it, and then they will modify themselves. The new gene will be part of their genetic makeup. So the plant's chromosome, the newly accepted DNA, will keep on replicating or multiply within the body of that new host. Okay, in the fig number 4, timifications causes tumor to develop onto the plant. Now the plant cells contain one new gene. If tumor cells are taken and cultured from that particular plant, the whole new plant can be grown from them containing the new genes, and now those transgenic plant gene will be able to resist or repel the tumor causing genes. That is how we can make the plant species immune to that particular gene. Next point over here is the genetically modified animals. They are basically those who are developed or who contain multiple genes into them, which we were looking for. So it is difficult to transfer new DNA into the eukaryotic cells of animals, but some attempts have already been made successful. Some of the most exciting and recent researchers have involved genetic engineering has been the work on inserting human genes into the tissue of sheep and cattle to produce the transgenic animals. Obviously, they would be the modified animals. Now, simply the first transgenic species was born. Okay, it was called as the trace, who produced the protein, the human protein alpha 1 antitrypsin. Okay, it produced the first ever produced transgenic protein was double A T in her milk. That protein is missing in some people, so we can take medication. We can use that milk as a medication that will be provided to those who are lacking in this double A T. So that contain double A T protein. So that milk is fine for such people to consume. It could be used as a medicine. The production of proteins using transgenic animals involves introducing a copy of a human gene which codes for the desired protein into the genetic material of an egg of a different animal species. Okay. Next thing would be as well as the gene for the specific protein. There is also a promoter sequence which makes sure the gene will be expressed in mammary gland of a lactating female. Now this gene will be straight away transmitted to the mammary gland of that female, so that even that sheep could produce double AT protein milk. That will be that could be used as for the sake of treatment basically. The fertilized egg, now the developed transgenic embryo, is then replaced inside the surrogate or substitute mother, is born and grows up to a maturity. When the animal is mature and start producing milk, the milk is harvested and purified so that the human protein could be extracted. So first we have to grow it, then we have to purify it to extract that gene which human needed as a treatment. So far, scientists have succeeded in producing transgenic sheep, cow, pigs, and certain animals. When we use large animals such as cow and sheep, large volumes of milk, and therefore human protein can be achieved. So in this way, we can increase the breeding or cloning. Again, same example of milk and meat. We can multiply them for the sake of human benefit. Here are some examples of drugs from the transgenic animals. They produce more than 20 different human proteins, and some of them are already in therapeutic use or being trialed. So factor number 7 or 9 are both important components of human blood clotting cascade. As blood clotting cascade is just like stairs, the way we move through the stairs one after the other, in the same way every factor is going to activate the next factor to it. So they can be missing in hemophilia, that is a blood disorder. So in that case, we can make person drink the transgenic milk by inducing these two factors into that milk, and that person could feel better, or even he would recover from the hemophilia. Or if not, if he's not recovering from it, he can live a better life though. It would trypsin is the protein produced by the the milk of such sheep contain up to 35 grams of human protein in every liter rare. Okay, it's a very high yield. The milk is very expensive. It costs several $1,000s per liter. Trials are in progress to make this treatment of them. Okay, so basically, in order to enhance that production of this protein, we have to invest. We have to invest into those trees. We have to enhance their production. That is why when a number of animals would grow higher, the cost of the milk would automatically come lower. Okay, so most of the transgenic sheep were destroyed when the company that produced them met financial difficulties. Obviously, they would fail to maintain their diet. They would fail to maintain their needs, but they were, or even the specific temperature that they were living. So they might have failed at that particular condition that result into the death of those sheep. Activated protein C for treating deep vein thrombosis is also being tested. So as we begin to understand the mystery of human genome in increasing detail, we should be able to identify the genetic sequence for every chemical in our body. If genetically modified organisms can make many of our faulty molecules for us, which we don't need, whether they are bacterial, plants, animals, or these things, so that could be higher in number. But meanwhile, body is doing to support us. Now we have reached that level of technology that we can remove the knocking out protein which we don't need. So that's it for this chapter. See you in the next time.